On this slide, we're looking at an animation of microtubule-based movement. What you're looking at here is the interior of a cell. So we're in the cytoplasm here, and there are organelles depicted in the background and cytoskeleton elements everywhere. The artist has drawn a microtubule right here. Here is a cargo containing some kind of material, and here is the motor protein. So I'm going to set this in motion so we can see what this looks like. Look at that motor protein. Does that not look like a cartoon? It looks silly. It looks like something that you would see in a silly cartoon. But this is literally what these look like. As you can see, as you watch the animation, it's the globular heads of the motor protein that are doing most of the work. It's the globular heads that are moving the most, and they are physically being sort of flung forward as each step is taken. Each one of those globular heads is sort of being thrown forward as each step is taken down the microtubule. On this slide, we're looking at the step-by-step process that is movement of kinesin motor proteins. So we expect a kinesin motor protein to take a step that's about 8 nanometers long, and it's capable of taking 100 steps per second. Isn't that amazing? That allows a kinesin to travel about 70 millimeters per day. So this isn't quite fast axonal transport here. For some kinesins, it's a slower process. But when we look at one kinesin and the way it moves, we can compare that to all motor proteins because they all move fairly similarly, although at different speeds. So if you begin in the process with the forward globular head binding to the microtubule, if you start right there, the foot, if you will, that's in front is going to land on the microtubule and it's going to bind. And it's going to bind specifically to the beta tubulin portion of the heterodimer. Now, when that happens, there's an ADP attached at that moment. And again, this will make sense as we talk through it. The ADP will be released, and an ATP, a brand new ATP, will come in and bind. The binding action of ATP to the motor protein is going to cause the protein to change shape. As we know, anytime you add something to a protein or take something away, you cause it to change shape. So as the new ATP comes in and binds, that will cause the region of the globular head called the neck linker to change shape. And it's that shape change to the neck linker region that throws the rear foot forward, the rear globular head forward. Now the trailing head, remember, the one that is now in the back is the one that's bound to that new ATP, and it will the ATP will be hydrolyzed, will be broken into ADP and phosphate, and the energy will be released. Now, because the ATP gets hydrolyzed, it's a different particle, it's a different molecule. That's going to cause, again, another shape change. The neck linker will change shape again. And that will bring it back to its original shape. Okay? Then the globular head will attach to the next beta tubulin. The ADP will be released, a new AT will bind, and the whole thing repeats. We have one of the heads binding to beta tubulin. A DP comes off, a TP comes on. That causes the neck linker to shift in its shape, which throws the rear foot forward. The ATP gets hydrolyzed into ADP. The neck linker changes shape back to its original form. The foot lands and binds to beta tubulin. The whole thing repeats. Let's take a look at this process step by step in another animation. Okay, in this animation, we're looking at the globular heads on the kinesin in purple. The neck linker regions are in red. Remember, we have a catalytic core inside each one of these feet, or globular heads. We have a neck linker region, and we have an ATP binding site. This right here represents the heavy chain region of the protein. We can't see the light chains, we can't see the cargo, they're out of our vision. But we can see the microtubule tract down at the bottom here, and you can see there are white portions to it and green portions. Those represent the two different types of tubulin, alpha tubulin and beta tubulin. I'm going to put the animation in motion now, and we're going to see what happens. So those are our globular heads with their core. Those are the neck linkers. There's the microtubule. What they're calling the coiled tether, those are the heavy chains. Now, ADP is currently bound. The foot is going to come down and bind to the beta tubulin right there. That's going to allow ADP to come off and ATP to come on. The binding of ATP to the foot is going to change the shape of the neck linker. Now it's turning yellow. And that shape change is going to throw the back foot forward. See that? Now, two things are going to happen at once. This ATP is going to get hydrolyzed, which will get the neck linker back to its original shape. Meanwhile, ADP comes off, ATP comes on, neck linker changes, foot gets thrown. ATP gets hydrolyzed, neck linker goes back, and the whole thing repeats. Binding, switch, shape change, throw. Shape change, binding, switch, throw. It's a remarkable process, and it's completely dependent upon the fact that proteins can change shape. And when they change shape, they change function. They do something different. And in this case, they move. So kinesins and dyneins are going to be moving in different directions. The kinesins move in an anterograde way, and the dyneins move in a retrograde fashion. Retrograde meaning towards the minus end of the microtubule. We said there are several types of kinesins, but there are two types of dyneins. There are cytoplasmic dyneins, and there are axonemaldines. Now, the cytoplasmic dyneins are the ones that we were talking about when we talked about the work done with the squid giant axon. They are moving, traveling within the cytoplasm. Think of those as being in the cell. The axonemaldines are the ones that live outside of the cell in cilia and flagella. We'll talk about those separately. The process of movement, the way the ATP is involved, the way the protein changes shape, the way the feet are thrown. It's very similar in kinesins and dyneins. When you look at cytoplasmic dyneins, they have an anatomy that's quite similar to what we saw in the kinesins. But unfortunately, you'll see there are a couple of differences. So they too have heavy chains and light chains, but you'll see there is no description of a globular head. And instead, there's what are referred to as intermediate chains. So some of the words sound the same, but they refer to different structures in a dynein. It's so unfortunate. The heavy chain terminology is used to describe the feet. The feet. The light chains are what's holding the cargo, so at least that's the same. And the intermediate chains are that middle tether, what we called heavy chains in a kinesin. Now, the other difference to note about the structure of dynein is that, unlike the kinesins, there are some accessory proteins involved with dyneins. There's a molecule called dynactin, and then there's what's called spectrin and ankyrin. Think of these accessory proteins as helping the dynein to do its job, particularly the job that the light chains do, which is holding on to the cargo. On this slide, we're looking at a drawing of a dynein in association with a microtubule. So the microtubule is drawn at the top here, again with the heterodimers represented by gold and yellow circles. Here's the dynein in brown. Here's the cargo down at the bottom. So this cargo is sitting inside of a vesicle, and the dynein is carrying it along the microtubule. Now, here are what we called globular heads in the kinesin molecule. In dynein, they're called heavy chains. Here are what we called the light chains in kinesin. They're also called light chains in dynein. They're what contacts the cargo. The coiled region in the middle was called heavy chain in kinesin. It's called intermediate chain in dynein. So this is cytoplasmic dynein. It's inside the cell, and it's going to carry cargo along the microtubule in a retrograde fashion towards the minus end. Over here in blue and in purple, these are those accessory proteins. So think of them as sort of being just a little extra help for the dynein. As you can see, they're sort of helping the light chain hold on to the cargo, and they're helping sort of keep the cargo at a certain distance from the microtubule so the dynein can walk, can do the movement that's required to deliver the cargo to its destination. On this slide, we're going to watch motor proteins as they travel along an axon. Now, what you're looking at here in the center of your view is the squid giant axon, and the bright areas are motor proteins that have been labeled so that they appear bright. Now I'm going to put the video into motion for us, and I want you to just look at what's happening. You can see little bright dots traveling along the length of that axon. Hold on just a moment and I'll replay it for us. Some of them are moving to the left, some of them are moving to the right. They're sort of walking past each other. This is actual microtubule-based movement occurring in real time along the length of this axon. Some of the motor proteins are traveling to the positive end of the microtubule. Those would be the kinesins. Some of them are moving towards the negative end of the microtubules. Those would be the dyneins. Lots and lots of cargo being transported along this axon. Microtubule-based movement of motor proteins is critical to the sorting, to the trafficking of protein in a cell. If you recall from our discussion of protein sorting, we talked about the role of the endomembrane system. We talked about how critical this interconnected pathway of membrane was to delivering protein to their destination in the cell. In this diagram, you're looking at the rough endoplasmic reticulum. Up here at the top, you can see the green ribosomes along its surface. We're looking at some transport vesicles in between. Here's the Golgi apparatus in purple. Here's the cell membrane. Now, what you're looking at right here is a microtubule organizing center. And inside of it, there's a pair of centrioles oriented perpendicular to each other. And all of the golden colored lines coming out of that centrosome, all of these are microtubules. Remember, microtubules grow out of the centrioles in all directions in 360 degrees, so they can reach any location in the cell. Now, if you think about the anatomy of this, if you think about the structure of this, remember that the negative end, the minus end, is going to be what faces the microtubule organizing center. The positive end is going to be away from the centrioles. So these microtubules all have their positive end at the plasma membrane. In comparison, these microtubules all have their plus end way up here, probably where the nucleus is located, or near to the nucleus anyway. So what that means is the motor protein that would be required to send a cargo from the Golgi apparatus to the plasma membrane for exocytosis, you would need a kinesin to do this, because you'd need a motor protein that travels in an anterograde fashion towards the plus end. In comparison, if the cell were trying to get a protein from the rough endoplasmic reticulum into the Golgi apparatus, and we've talked about why that would be necessary for protein processing, you would need a dynein to do that work, because the plus end of the microtubule is up here. So you would need a motor protein that's capable of traveling retrograde towards the minus end, which is over here. You would need a dynein, a cytoplasmic dynein, to carry cargo from the rough ER down into the Golgi apparatus. So both motor proteins are critical for moving things along and throughout the endomembrane system. We're going to switch gears here and leave cytoplasmic dynein and instead take a look at axonemaldine. Now, I said axonemaldine is the type of dynein that we find in the external cellular structures, the cilia and the flagella. And what makes axonemaldine very different from cytoplasmic dynein is that it doesn't walk along the length of microtubules, but instead it's anchored and it moves in place. Let's think for a minute about the structure of cilia. Cilia are the external hair-like structures that can be found on certain cells. They are short and they are thin, and there are many of them when they are present. So about 2 to 10 micrometers in length, only about 1/4 of a micrometer wide. You find cilia in unicellular eukaryotic organisms, and they do the job of not only helping the cell move through its environment, or what we would call locomotion, but they also help that primitive eukaryotic organism eat, because they help to sweep food particles towards whatever opening the cell uses to bring its food in. In multicellular eukaryotic organisms, cilia play a very different role. Cilia are instead moving substances past a cell. So if you think about humans, for example, you can find cilia in humans in the respiratory tract. You can find it in the trachea. You can find cilia lining the cells that line the trachea. Each individual epithelial cell inside the trachea has hundreds of cilia on it. And those cilia are working together in a sweeping motion to carry both mucus and the dust and little particulate matter that gets stuck in the mucus up and out of the area of the lungs. So those cilia beat together and they carry all of that debris up towards the back of the throat, so it can either be swallowed or it can be spit out, or whatever it is that the person does with it. You can really see the importance of cilia in action when you look at a person who smokes cigarettes. You've probably heard a smoker cough before. They have a very distinctive cough. It's a very deep and very wet cough. And the reason that smokers cough so much is because cigarette smoke actually destroys cilia. So the normal cilia that does all that work for us is gone in a cigarette smoker or a person who smokes any kind of material and draws smoke down into their trachea every day. The cilia are destroyed by smoke, and so the mucus and the particles within it have no way to come up and out of the trachea, and instead start to settle down into the lung tissue. And that's what causes those people to cough. That's another mechanism to get that mucus up and out. Human beings also have cilia, at least females, inside of the oviduct. Remember, the oviduct is the little tube that connects the ovary to the uterus. And inside the oviduct, there are cilia there that help to transport the egg from the ovary where it's released down towards the part of the oviduct where it will be fertilized by the sperm. When cilia beat, when cilia move, they move in unison, and they have a very distinct two-part motion. They have what we call a power stroke and then a recovery stroke. And the motion involved, the motion of the power stroke and the recovery stroke, is created because of the dynein, the axonemaldine that is anchored in the cilia, is what is providing the power stroke and the recovery stroke. On this slide, up at the top, you're looking at an EM image of a group of cilia inside of the trachea. So each one of these little hair-like projections is a cilia, and together they work to sweep things away from the surface of a cell, if you're talking about a multicellular organism, or they help the cell move, and they help the cell eat, if you're talking about a primitive single-celled organism. In the graph in the center of the slide, we are looking at how cilia move. And remember, all of them move in unison on the surface of a cell. On the x-axis here, we're watching time move forward, and we can see a drawing of a cilium right here. So the power stroke is the forward sort of bending over of the cilia, and then in the recovery stroke they stand back up. So they sort of fling themselves forward and then they pull themselves back up. Power stroke, recovery stroke. This is motion, this is mechanical motion, movement, and it's provided by the actual motor proteins that are in the cilia, the axonemaldine molecules. At the bottom of the slide here, there's another EM image, but this one is showing a flagellum, and we'll talk about those separately. Axonemaldine is providing movement in place because the dynein motor proteins are anchored inside these extracellular structures. They're also providing the motion for flagella, but it's a little more complicated in a flagellum because flagella are enormous compared to cilia. A flagellum can be up to 200 micrometers in length, and cells can have anywhere from 1 to 10 flagella if they have flagella. Now, in a unicellular eukaryotic organism, a primitive eukaryotic organism, the flagella are there for locomotion. The purpose is to help that cell move through its environment. The motion that a flagellum makes looks very different from cilia. Instead of having the power stroke forward and the recovery stroke backwards, flagella actually sort of undulate. They sort of wave and they rotate in 360 degrees. Very complex movement. And they need a lot of motor protein to achieve that movement. So they are anchored along the length of the flagellum. There is a cell in the human body that has a flagellum, and that cell, of course, is the sperm cell. Sperm cells have to travel a tremendous distance from where they are deposited in the female, in the area of the cervix, all the way up through the uterus and into the oviduct, where they meet the egg if it's there. So the flagella on sperm cells allow them to swim that tremendous distance. We're going to look at a short video here to take a look at cilia and flagella in motion. We already saw some of the motion of cilia in our laboratory exercise when we were working with tetrahymena. If you recall, the tetrahymena have cilia, and like all primitive eukaryotes, they use that cilia for two different things. They use it to move through the environment, and they use it to sort of sweep food particles towards their oral groove. You're looking at another primitive eukaryotic organism on this slide in this video. This one happens to have flagella. There are two of them on this little cell. Now, this cell has been exposed to that material called protoslow that I mentioned in the laboratory. So it's been slowed down. But you can see that the flagella are moving. They're waving, they're undulating. This is another cell here. It too has flagella on it, and the flagella are undulating. This one has flagella up at this side. You can really see that the positioning of the flagella and the number of flagella, they're up here, are what determines exactly how the cell will move. This one's rotating as it moves forward. Here's another one with several flagella. Again, it's sitting in protoslow, so it's kind of hard to see the natural movement. But flagella are going to wave and rotate. These are paramecia. They have an oral groove similar to what we saw in the tetrahymena. They don't have flagella, they have cilia. But notice how rapidly and how carefully they can move through their environment. Here's that oral groove where food particles can be taken in, and you can see the cilia waving along the outside of that cell. All of this motion that you're seeing is provided by Axonemaldine inside the cilia and inside the flagella. So let's take a moment now and think about the anatomy of the cilia and the anatomy of the flagella, particularly in terms of the location of the axonemaldine. Inside both cilia and flagella, there is a structure called an axoneme. And the axoneme is attached to a structure called the basal body. The basal body is what connects the cilium or the flagellum to the membrane. So if you look at the basal body first, you're going to see 9 sets of triplet microtubules around a hollow core. Does that sound familiar to you? It sounds an awful lot like a centriole. It sounds just like a centriole, in fact. So this basal body structure with these 9 triplet sets is connected to the membrane. And then if you start to go outward from there, you'll encounter the axoneme, which also has 9 sets of microtubules in it, but now they're doublets. 9 sets of 2 microtubules. And rather than surrounding a hollow core, they surround a central pair of microtubules. Now this is where the dynein comes in. I'll show you an image of this in a minute, but you have to imagine that you've got this central pair of microtubules, and then you've got a tube around it. Think of a bicycle tire here. The dynein is going to be the spokes of the tire. The dynein is going to connect the outer rim to the inner central structure of the tire. The dynein are often referred to as side arms or spokes, radial spokes, just like we would think of the spokes in a tire. On this slide, you're looking at a cross section through a cilium or a flagellum, and you're also looking at a longitudinal view, and you're looking at an actual electron microscope image that was taken of a cross section. So you can see in this image there's membrane here, and then there are these doublet or twin rings, and there are 9 sets of them around a circle. Okay, so these are those microtubule structures in pairs. In the center, there's another pair, a central pair, and what connects them are these spokes. These spokes are axonemaldine. Now look at it as it actually appears under the electron microscope. You can see the doublet microtubules. 1, 2, 1, 2, 1, 2, 1, 2. You can see the pair in the center, right here. And then these right here. These are the dynein motor proteins, acting as the spokes of the tire. Now, the cilia, again, are much, much shorter than the flagella, so while they have the same basic structure, the flagella is a lot longer. So the flagella just is full of much, much more dynein because it needs more dynein to provide movement for the longer length of the flagellum. On this slide, you're looking at some more EM images. This is a flagellum longitudinally cut, And then here you can see it in cross section, but at different levels. So you have to imagine that there's cell membrane down here where this flagellum has been attached. So that means the basal body is right here. And remember, in the basal body, I know it's hard to see, but in the basal body there are triplet microtubules arranged around a hollow core. And then as you move upward, you get into the axonemal region, and then we move into doublet microtubules around a central pair. And this is where the dynein is located. The spokes are located. The dynein, the axonemaldine, is what provides the power for cilia and flagella to move.